So good morning. Please try to find your seat or a new seat if that's easier. So we would like to take the next step in this important dialogue that started this morning on the battlefield. And then we moved closer to the academia and governmental funding and R&D research opportunities here. And now we'll take a step into discussing improving efficiency in drug development. And I'm very pleased to be able to monitor this discussion, moderate this discussion, and being joined here by some really leading voices in this area of translating discovery to important medicines. I think what we've seen coming now is a real unique opportunity of convergence of advances in science and technology that can allow us to take on bigger challenges and aspire for more impactful, transformational medicines, vaccines, and healthcare solutions. We've had a decade of more incremental advances in many areas, but they are still important. We build on that knowledge, but now can hopefully reach for some more transformative advances that can really change outcomes and lives for patients. As we see on these positive opportunities, we have listed a couple of key aspects. We heard Professor Fogelstein speak about the classification of cancer based on more molecular and genetic abnormalities rather than traditional physical and histochemical. A new taxonomy of disease is emerging that really allow us to understand the molecular basis not only of cancer, but gradually of other diseases, immune diseases, metabolic diseases, and possibly at a functional level, at the circuitry level of psychiatric, and also at neuro-regional level of many degenerative diseases. New enabling technology, particularly great progress in high-throughput biology, informatics to manage huge data, allow us to extract information in an unprecedented manner. And as we start to understand these pathways that are linking disease processes to unhealthy cellular outcomes, we are increasing our ability to design highly sophisticated small molecules by rational drug design, co-crystallization of targets and compounds, but also in biologicals, we're able to make more sophisticated antibodies, proteins, and vaccines than ever. We've seen the rise of precision medicine that lends to the notion that better selecting patients will allow us to increase widely the therapeutic window and allow benefit to happen to the right patients and reduce safety issues or treat those that don't really gain. At the same time, as we have this opportunity to maybe in decades change how we do clinical development, there are also really important tools such as large patient registers with longitudinal phenotypic and increasingly also genotypic data that can allow us to monitor patients in the real world and also in real time see how new medicines are providing both benefit and risk to patients. This opportunity has forced, for good reason and for challenges, new type of collaborations between government, industry, academia, and I think also recently, really important, the patient engagement. patient foundations provide a unique insight to the needs and priorities of patients, and they can make things happen in the interfaces between stakeholders when the dialogue doesn't move the topic forward fast enough. So on this lower left corner, I think we see a number of important components that really can transform how we as an industry can support innovation into important medicines. But there are also a number of continuous challenging pressures. In the regulatory environment, there are still a lot of complexity, hurdles that makes it difficult to bring science forward in diseases such as cardiovascular, diabetes, obesity, and many of the neurodegenerative diseases, requiring very, very large clinical studies, and particularly for early intervention, still uncertainty about which markers can be agreeable to serve as surrogate endpoint. So although we are pleased and have welcomed the great guidance in the regulatory arena in oncology and rare disease, ability to, through breakthrough designation, get more interactive dialogue and accelerated approval to bring important medicine faster to patients, there are many areas where the same type of spirit needs to lead to new ways of developing medicines under obviously safe and thoughtful conditions. And once we get those medicines that may have thousands and tens of thousands of patients enrolled in clinical trials to approval, we often are faced with very large post-approval commitment that will, on one hand, take many, many years before the learning come back. And it's linked to a large spend that doesn't allow the industry to refocus where our resources can open up new doors, new avenues for breakthroughs. And many of those commitments are based on historical, very large, randomized clinical trials. While we start to see, as you noted in the left-hand corner, the ability to monitor patients in real time with registers. So we still haven't got that dialogue worked out fully, how we can get a balance between the traditional and the modern technology to monitor new medicines and learn about them. So on one hand, I think you will hear from this panel of colleagues about we see positive signs on industry productivity, but there are also concerns, and there are still far too many late-stage failure rates in phase 3 that are really what can threaten the return of investment for this industry and the support for translating science into important medicines and vaccines. So I will soon ask each of the colleagues here in the panel to share some of their thoughts about this environment of opportunities and challenges and exemplify with the learnings from their R&D areas. I'm pleased to have here Jeff Leiden, cardiologist, professor at Harvard, president previously of Abbott Laboratories, and during recent years, CEO of Vertex. Elliott Sigal here next to me, MD, PhD, experience from biotech industry, worked at UCSF, and I think for 17 years been a leader at Bristol Myers in R&D, and the last 9, where Elliott was the president of the BMS laboratories, was a period that saw some 10 medicines come to approval in a variety of different diseases. So I certainly look forward for listening to your experiences. And I think you will share with us one particular interesting case in oncology. And Jan Lundberg, renowned pharmacologist, led for a decade, discovered AstraZeneca, and since the last couple of years, now leading the research laboratories at Lilly, taking on the new challenges in this environment that we will be discussing. So I'll start with asking Jeff to share some of your experiences here.
Yeah, thank you, Mikael. I thought I'd talk a little bit with you about Vertex's experience with cystic fibrosis, and these predate me, so I can look at them both as a participant and a historian a little bit. And the reason that I think they're interesting, at least from a personal standpoint, is as I've watched this 10-year research and development initiative, I've learned a lot of things that changed my mind about the best ways to develop these kinds of breakthrough drugs. And I thought I'd just share a few of them with you because I think they are relevant to how we're going to think about a new paradigm of drug development going forward. The first thing I would say about these efforts, and Mikael mentioned this earlier, is that they really represented a very unique collaboration between academia, industry, and patients and patient advocacy organizations. And it's relevant to what we were talking about in the last panel, I think, because I view this really as an ecosystem with multiple parts. And if you damage any one of the parts of the ecosystem, whether that's decreased NIH funding for basic research, or whether it's trying to damage the interaction between industry and academia, or whether it's trying to separate how companies interact with patients, you'll actually destroy the whole ecosystem. And I think what I'll tell you about wouldn't have been possible. And so I think it's important to see it as this unique collaboration because it drove things forward very fast. The second thing I would say is, this started off in academia. I was actually at the University of Michigan back in 1989 when Francis Collins and Lapchi Choi discovered the CFTR gene, which is the gene underlying all of the patients with CF. So it was very much a basic academic discovery. And the first thing, which was sort of depressing news that was realized very quickly, was there were 1,900 different mutations in the CFTR gene that could cause CF. And from a drug developer standpoint and a precision medicine standpoint, that was initially a pretty depressing finding. How are you possibly going to develop medicines that could address all of these different mutations? So the second unique insight really came from industry and academic investigators, including some Vertex scientists, who understood that you could bucket these mutations into a couple of groups functionally. So there were mutations in which the protein, the CFTR channel, got to the surface of the cell but wouldn't open. And they were a set of mutations. There was another set of mutations in which the protein misfolded and got degraded and never made it to the surface of the cell. And when you actually bucketed things that way, you began to understand that from a cell biology standpoint, it might be possible to address the disease with just 2 or 3 or 4 different kinds of medicines. And that was really the other finding. That was the beginning of precision medicine. And so it allowed our scientists to develop some unique screens. And these were phenotypic screens, interestingly, sort of the old days of drug discovery. So even though we had the gene, we had the protein, we didn't do classic biochemical screening. We actually used cells from patients' lungs who had CF with these different bucketed mutations and developed high-throughput phenotypic screens, which when you look back in retrospect was the absolute secret of success. Those screens read out molecules very, very differently than transfected cells would have, or that biochemical assays would have, or that artificial lipid membranes would have. And so very quickly, within just a few years, our scientists out in San Diego were able to utilize those screens to discover a series of molecules that were precisely targeted to these different buckets of disease. And because of that, they were then able to take them into the clinic in very restricted populations. If we had taken our first medicine, Kalydeco, into the clinic in all CF patients, it would have failed. And yet, because we took it into the clinic in a very specific mutation, which the receptor got to the surface but wouldn't open, it actually allowed us to have a very high success rate, even though that was only 4% of the total patient population. So that was sort of lesson number 3, is the importance of understanding the genetics and the phenotype at the same time when you're thinking about a drug development effort. The last part that I'll talk about now, Mikael, is the collaboration with patients, because once we had these medicines, as you may know, the CF Foundation had organized all the 35,000 patients or so with CF in the US. And they actually had them all genotyped, and they were all being treated in a series of just 270 centers in the US. And so once we had medicines that we could test, we could enroll these clinical trials literally in weeks, and we could get them done in 6 months to a year. And so we were able to go really start to finish in 4 years to get the Kalydeco drug development program done and to submit that application to the FDA with Kalydeco, the first of these medicines. And then, we were talking about the regulatory environment. This was a case in which the FDA, I think, had a very clear understanding of what we were doing. The patients had talked with them pretty clearly about the importance, and they actually approved this application in less than 3 months from the time of submission. So in many ways, I think it serves as a real paradigm for a new way to accelerate both the discovery and the development process. And one of the questions I always get asked is, well, CF is sort of an outlier, right? You really couldn't do that in other diseases. But in fact, we could list 20 diseases which are quite similar in terms of how one might think about the genetics and the cell biology, in which one might think about collaborating with these patient groups who are prepared to get trials, and I think in which the FDA acknowledges that such breakthrough therapies would move very quickly through their system. So it really is an interesting paradigm to look back on and take some lessons from, and I think I take a lot of encouragement from it because I think it describes a way that we may be able to move forward in many diseases.
Thank you, and that's such an important example for how science can help few patients that suffer greatly. John, you wanted to share an experience in another area.
Yeah, we recently celebrated at Lilly the 25th anniversary of starting our Alzheimer's program. And of course, the conclusion was that we haven't delivered any medicine in 25 years of research. But it's still, I think, important to understand that we have made a lot of progress in the area, in academia and in the pharmaceutical industry. Unfortunately, all of the phase 3 trials so far, haven't really worked in influencing disease progression. We have therapies for, I think, temporary symptomatic improvement, but the large challenge still remains. The good thing, though, here in this area is that we are increasingly understanding disease pathophysiology, and I think the evidence that amyloid A-beta peptides are involved, at least in the early stages of disease, are increasingly strong, based then on human genetics, both then gain and loss of function. And of course, if you're lucky, perhaps you're one of the few Scandinavians then that are protected against Alzheimer's disease because you have a mutation in the base cleavage site of the amyloid precursor protein. But that's not the case for most of us, unfortunately. And the thing that we, I think, are seeing happening is that there are new ways then of addressing amyloid and tau pathology. And I think perhaps the most important thing that has happened recently is also the ability to see these changes in the living human brain. Just a few years ago, the diagnosis of Alzheimer's was actually made at autopsy, because it was the histology of amyloid plaques and tau tangles that was the finite diagnosis. Now we can see both amyloid and tau in the living human brain. We can see how it changes with time, and potentially we can use it to select patients for our trials, and as I will exemplify also then, perhaps to use as surrogate markers, particularly the tau component, and which is directly linked to neurodegeneration, as it, at least preliminary data suggests. So is there any agent that has shown any benefits in a large phase 3 trial? Not on primary endpoints, which the regulators want. They want the endpoints both on cognition and function together. You probably heard that the Lilly agent sulanesimab had a secondary endpoint with pooled data in mild Alzheimer patients that actually was positive. And here there was a 34% reduction in the progression of cognitive decline than at 18 months of treatment. And we find this extremely encouraging and have started the next big phase 3 trial, trying then to learn from this and to optimize the next trial, which is called Expedition 3, by using imaging first to select patients that really have amyloid in their brain. And we know now, I think, that 20-30% of patients, even in the mild category of dementia, does not have amyloid. So why do we treat them with anti-amyloid agents? And we also know that these people do not really progress very quickly, so we should really exclude them. But we are also increased the size of the trial. We are only studying mild, not moderate, and we are using also geographies and sites which have higher reproducibility of how then to interpret these cognition and functional parameters. So hopefully within 2-3 years, we can show that we could confirm these data, which I think could be a very important step. And finally, sulanesimab had what we consider acceptable safety, which has been a dilemma for some of the other antibodies against the amyloid in humans. The tau story, I think, is one of the recent most interesting areas, because tau is really the end-stage sign then of neurodegeneration. And the way now to look at tau has in the past been then CSF samples and looking at biochemically measuring tau levels. Now we can image tau where we can look in changes in specific brain regions, which seems to correlate very well to symptom loss. And we can also follow the decline then of the tau imaging agent in relation to how the disease progresses. And in fact, there is a very good correlation between cognitive decline and the accumulation of tau in the brain, which is quite different from amyloid, which doesn't have that property. The other thing about tau, I think, is some breaking science from academia suggesting that Alzheimer's perhaps could be a prion disease, because some of these misfolded proteins like tau, if you inject them in, at least in rats, they spread throughout the brain, just like prions have been described to do then in mad cow disease. And we and others have recently shown that you can actually give antibodies peripherally and prevent this spread of tau in the brain. So that could be another therapeutic angle then to the whole era of Alzheimer's disease. So in my view, we have made progress. I think we increasingly recognize who should we study. Well, we should study the patients that have amyloid plaques in their brain, and we should do it as early as possible to have then the ability to influence it. For how long should we treat them? And here it's a dilemma, because Alzheimer's disease in the early stages is a very slowly progressing disease in most patients. And so far, most studies have been 18 months, but perhaps we need to extend these studies, which makes it more complex from a cost and regulatory perspective. Which therapeutic agent? That's, of course, one of the key questions here. Is it an antibody? Is it a BACE inhibitor, an oral agent, or is it a combination of both? And we and others have recently seen some very intriguing data of combination treatments, because it may not be enough just to block then the new formation of amyloid, but you have to remove the plaques that you already have in the brain. And how do you do that? Because when you diagnose these patients, they already have plaque, which comes as an early stage then in this whole disease. And the tau era, like I said, is opening up with antibodies, and there are also new ways with oral agents that potentially could induce then changes in tau. So my summary is also that we need to continue our interactions with the regulators, including the FDA, where we are having debates about what's the most appropriate cognitive and functional endpoints, and could the tau imaging changes, for instance, be used as a surrogate marker? Clearly, we need to show that then by studying the parallel endpoints with cognition and function and the imaging to see if they correlate. And in the end, of course, the 25 years of research as Lilly has then, of course, to succeed within the next 5 years, I hope. So that's my message. Thank you.
Thank you, John. And I think, Elliott, you will take us back to the early intriguing story that Bert Fogelstein concluded with, the immunotherapy meeting the targeted therapy and interacting at the right time in the patients.
Yes, before I do that, I'd just like to say that I've been doing a lot of reflection about the industry recently. I retired in July, so I've had a lot more time to think. I should have done that earlier. The thinking part, not the retirement part. And I'm very proud of this industry. So despite all the challenges and the concerns that you hear, I would like to make sure that the younger people in the audience that are dedicated to this understand what a privilege it is to make medicines for patients and to do so in the organizations throughout the ecosystem we belong in. I will say that there's been a big transformation in R&D productivity at Bristol Myers Squibb. This has been written about. This is has independent metrics from the period of before 2002 and then afterwards, and no one person's responsible for that. And I can't tell you exactly what happened because it's not a controlled experiment and I'm talking to a bunch of scientists, but I will give you my thoughts because I think some of them are extrapolatable to different organizations. And a lot of it has to do with culture, because after all, any leader is responsible in the end for creating the environment to allow good things to happen in an organization. I would say at the outset, never let a crisis go wasted is the phrase. And we had our share of crises in 2002. To refresh your memory of a little history, we had extreme patent exclusivity losses. We had a failed phase 3 antihypertensive drug. We had licensed in Erbitux from mClone, received promptly a refusal to file the first time from the FDA. There was a scandal involving headlines because Martha Stewart was involved. There was insider trading, a congressional investigation, channel stuffing, and that was all within my first 2 months. And I learned something from talking to the young clinical scientists working on Erbitux, because I was obviously worried about the morale. We had 20% attrition. We had a lot of concern about whether we could do anything internally, what they were writing about the company, our R&D capabilities, the future. And Marty Burkhofer, one of the oncologists on the Erbitux trial, said, you know, Elliott, this is not about any of that. It is about the patients. And I realized, as Dr. Vagelos said, the industry at times has indeed lost its way. And always the North Star to come back to that will work is patient focus and all of the ramifications. And George Merck, to paraphrase him, was always right. I don't in saying that if you do what's right for patients, the profits will follow. And so I disagree a little bit with Rob Califf, and almost never disagree with him too strongly. Finances is not the only aspect in any decision making, and when the balance gets out of whack, you make bad decisions. So we developed a strategy that focused on the patient and focused on unmet need. And that's the North Star. Unmet medical need allows you to get the right science to the problem. It allows a decent business model. It helps with regulatory hurdles. It helps with the payer situation. And you are allowed to tap into why people come to our organizations to work. I'll say a few things about research and in development, just some highlights of what were put into place under this overarching paradigm, and then I'll give the example of immuno-oncology because it exemplifies that. In research, we froze and actually slightly downsized the capacity in-house to 50% of what we knew an organization our size needed. We did that to maintain small groups, core competencies, and develop a culture of wanting to bring in innovation from the outside. It's clear that non-invented here is a syndrome in many organizations. But if you can align people to the overall collective efforts of bringing in things for their area and admit from the beginning that we have to go outside, not in a crisis, but all the time, you can achieve an effective discovery operation that's smaller and more nimble. In the development side, I was a researcher that transferred into development, and I must say I always thought that development was quite rote. It was engineering. It was turn the crank. And I quickly learned that there is a lot of science and a lot of medical science in figuring out the right clinical development program. And you can't always look at the wish lists that might come from commercial or other sectors and replace that science. You can only do what the science tells you. There's no other industry where in development you're not just constructing something to match original specs, you're actually continuing to discover. The final thing is with those three principles of a small discovery operation, an attitude to go outside and to follow the science, I'll go to the well-known Medarex example of immuno-oncology and the approval last year of Yervoy, which was honored with the Galien Award for biotechnology product last year. It was 100 years ago, actually, that a physician in New York, Dr. Coley, noticed that his patients with sarcoma and a throat infection seemed to have, when the fever resolved, a regression of some of the tumor. And for many years, people tried to harness the immune system. There is an ecosystem with every drug that's invented that involves a variety of organizations, include academia, the government, biotech companies, small efforts, and big companies. In this case, a biotech company was bought by Bristol. They later discovered and elucidated the B7 pathway of T cells. They discovered the functions of CTLA-4, the late appearing antigen on cytotoxic T cells, but they got the function wrong. And Jim Allison elucidated the function that blocking the receptor actually unmasked what was a shield that the tumors created against the immune system. And if you took that shield away by blocking CTLA-4, you could unleash the immune system. And we saw patients that, like shown earlier, not only have their tumors totally dissolved, but that was a durable response. Other companies were in this race, but they didn't follow the science, in my opinion. Classically, the chemotherapeutic agents you expect to shrink tumors. The physicians we are working with on the outside and our clinical scientists realize that if you're eliciting an immune response, you're going to see tumors swell up first before they get better. And people that missed that signal and carried the science forward from the preclinical side into the clinic and listened to clinical signals as an experiment stayed with that program and found a small number of patients that had durable response. And we saw what I've never seen in my career in terms of a solid tumor KM curve, Kaplan-Meier survival curve. Separation, which we see and always get excited about and leads to approval, but we did not see any convergence over 4 to 5 years. In solid tumors, in my generation, I don't believe we've seen a KM curve that remains with daylight, even a small sliver of daylight, for 4 to 5 years. And now recent data presented suggests that we can start talking about functional cures in this field. We can improve the number of patients that experience this by looking at other mechanisms like anti-PD-1 or combining PD-1, CTLA-4, or other modalities. The elements of collaboration, the ecosystem, not taking everything that's invented inside and following the science and development are all illustrative in that example. And I think the culture of the patient focus and unmet medical need is key in our industry and must be restored.
Thank you, Elliott. That was a really nice summary of many years. Great work there. So before I open up, I wanted just to build on my the key questions raised by the panelists and bring you back to this balance I have on this slide and just share how we have been working on capitalizing maximally on the opportunities and try to in some way mitigate the pressures in Pfizer over the last couple of years. So similarly, as you heard from my 3 industry colleagues, we have been faced with productivity challenges and embarked about 3 years ago on a turnaround journey, trying to reinvent the way we discover and develop drugs. And a focus and you know, a key, some pieces, as you will hear, are similar between how we as leaders and as companies are trying to take on these challenges, and some are different. And of course, we need a diversity of approaches and learn from the ecosystem. So we clearly sharpened our focus on particular diseases, mechanisms, where we thought that the new taxonomy of disease, the deeper insight in human biology would be helpful, and deprioritized other areas which were based on very solid, more traditional animal work, which we felt had great challenges to translate into early signals of efficacy. If you look at the balance, it really involves an ecosystem from science to regulatory, and the outer end, of course, once we get the drugs approved, are the stakeholders willing to pay for these drugs? So we wanted scientists to feel they are equipped with a more holistic toolbox. And for us, it has been very important to bring the curiosity of scientists together with equipping them with glasses, a lens that allowed them to put on a kind of a perspective of a business acumen that really builds that bridge, not only from the the bench to the bedside, but all the way to understanding regulators and to the forces in the marketplace. The second point relates to precision medicine, and you heard some fascinating stories, of course, from the cystic fibrosis field. One example that I share from us in Pfizer was Xalkori, a small molecule drug designed as a kinase inhibitor that were antagonizing a target called ALK. And as we were developing that drug, some breaking science showed a rare translocation occurred in lung cancer in 5% of the patients. And as we started with diagnostics to focus on those patients, you could see these remarkable responses, response rates of about 60%. Very much that same feeling of being able to change lives and outcomes of patients, as you heard Elliott speak about. But unfortunately, we also learned, as we heard from Bert Fogelstein, that as we deliver that remarkable improvement for patients, and for some of them it may last 1 or 2 years, eventually for monotherapy, the tumors will relapse in the great majority of patients. And obviously we embarked on now translating the science around resistance to new medicines that I think in the next couple of years will come into those patients, and hopefully a combination of our drugs, maybe combination with medicines like you heard from in the immunotherapy field, will eventually bring cure to many of these patients. And that just underlines the third point about greater collaboration. And in the last couple of years, we wanted really to look at a new way to collaborate with academia. And we launched what we call Centers for Therapeutic Innovation, in which we partnered in almost integrated project teams with scientists and physicians at academic medical centers, asking them to propose the project that they felt had the best chance to translate into human impact in a joint steering committee, then deploying our ability to design drugs and really opening all our libraries, whether for small or large molecules, and translating that into as rapid as possible proof of mechanism in the academic medical centers. And we actually have here in the Alexandria Research Center, one of our CTIs, 1 out of 4, I think Jean Megram is actually sitting here, who leads that center. And we hope that would serve as an example how you can accomplish much more when you collaborate earlier in a more interactive and dynamic manner, and try to build trust by early sharing of information in a obviously controlled but trustful manner. So ending with those couple of examples that you've heard from 4 different companies, we would like to open up also for discussion. And I think we have one first question here. Is there a microphone here?
Good morning, my name is Silar Voros, and on behalf of Global Genomics Group, I would like to congratulate the foundation of putting this fantastic meeting together. Our mission at Global Genomics Group, as many of yours, is the discovery of new compounds. And I was very delighted in Dr. Vogelstein's lecture to show, when he was showing the roadmap of combining advanced imaging with a lot of the different omics. So what we're doing in the context of Global Genomics Group is in the context of cardiometabolic disease, large discovery project in up to 10,000 patients where we do phenotyping with advanced imaging and then do whole genome sequencing, epigenomics, proteomics, metabolomics, glycomics, and essentially all the next generation omics that you can imagine today. So I have 2 questions related to that for the panel. One is, in the kind of now the post-GWAS era, where do you see genomics fit into that discovery space, and specifically what is the role of whole genome sequencing in that? And the second question is, where do you, in this new strategy of really going after the orphan drugs, orphan diseases, orphan strategies, where do you see the role of biomarkers fit in, and do you see those sit within pharma, within the diagnostic companies, or do you see emerging of those 2?
So if I could ask Jeff to take on the orphan disease question and Elliott oncology, and then I have a question that came in that will fit for Jan here.
Okay.
You want to start? Yeah.
Yeah, let me try to talk about the biomarkers, and it's something we've thought a lot about because it is very relevant to our cystic fibrosis work. These drugs are getting approved on the basis of changes in FEV1, which is a fairly crude pulmonary marker of really mid to large size airway resistance, which isn't really the problem in CF, although it's a sort of side effect. And so we've spent a lot of time thinking about developing new biomarkers that reflect the systemic effects of the drug. For instance, these patients have malabsorption. They gain a lot of weight after they're put on our drug. They take a lot of pancreatic enzymes. They don't need those pancreatic enzymes. But more importantly, we've been interested in better imaging technologies to look at the lung. And so we've used things like lung clearance index, mucociliary transport, which you can do in an imaging approach. And what's remarkable is all those turn out to be much better indicators of the patient response when you actually see how they're doing and correlate that with a biomarker or an imaging marker. The challenge is how to get those through the regulatory pathway, because of course none of them are validated regulatory approaches. And so we've been spending a lot of time as we go through our initial set of trials with Kalydeco and now with combination therapy for the more complex mutations, putting those into the trial to try to validate them so that the next time around, and frankly when other companies come along behind us, they'll be able to use these novel kinds of biomarkers. The only other thing I'd say, which I think is a really interesting development that's going on in the industry, is we've always done clinical development by having patients come to their physician's office and getting their markers and their bloods and everything else measured. I think we're definitely seeing a movement in which we're asking patients to collect real-time data at home in a very, very different way. So for instance, we've given patients Bluetooth spirometers that they can use to do their FEV1 at home. It goes right to their cell phone, it comes to us. And what we see is that data is often much more reliable, as you might expect, than a once every 3-month visit to the physician where they do an FEV1. And so I think one of the things we need to think about from a regulatory standpoint, but also from a reimbursement standpoint, is having the patients work with us to collect this real-time data using some of the new technologies that are available to us.
Thank you, Jeff.
I'll just add a few comments on your question about genetics and drug discovery and development. Clearly, it remains a major challenge to start off with the right target. Target validation in human remains, I think, a major bottleneck. There's nothing like having validation in the human through genetics, in my opinion. And certainly there's some well-publicized recent examples, not fully borne out yet with late-stage studies, but coming. Helen Hobbs's work at PCSK9 in cardiovascular disease and lipid metabolism. And one of the reasons we were very excited in diabetes with inhibiting the sodium glucose transporter, the second isoform in the kidney, to flush out glucose, fluid, lower blood pressure, weight, by reduction of calories, was the fact that there were a small number of patients that, at least the heterozygotes, seemed not to have any safety issues. And that was the question about targeting the kidney in diabetes. And they also had benign glycosuria, which is commonly picked up in people that are enlisting in the Army, and there's nothing else wrong but dumping too much sugar because of a defect in SGLT2. So with better techniques, genome-wide association studies, faster and cheaper sequencing, we certainly expect and hope, I think primarily in the academic community, that we get more validation and elucidation of biology. And we were talking earlier about partnerships. I think it's critical that we rely on the production of human disease insight out of academic institutions, and this interface at medical centers between the basic science and the early clinical studies.
I just wanted to build on Elliott's discussion here. When it comes to use of genetics or whole genome sequencing to select the right patients, we are still struggling in oncology with a very traditional path for approvals, where when you have a targeted therapy, you have to develop a unique diagnostic. And as you often learn more about your drug during the development or in the marketplace, you find additional markers, but you can't really use them as part of the way how a drug is labeled and promoted unless there is a fully validated diagnostic test and very large clinical studies run on them. Although in most academic hospitals, they use this technology routinely. So to see that kind of one diagnostic pad with one drug replaced by the use of, at least interim, a panel of the maybe most 50 relevant genes in lung cancer, or ideally whole genome sequencing, and just through a validated methodology, you can quickly evolve the drug to more effective use, whether in clinical development or in the marketplace, than a very sequential, slow way of doing it. So I do hope that could happen, bringing diagnostic and drug together. It could really transform the journey here. We've got one question that I wanted really to have John weigh in on concerning industry-wide collaboration. One of them is Transcelerate Biopharma, which is involved in drug development. And the question is whether and how these type of initiatives impacts your organization. And John, as you speak about Transcelerate Biopharma and drug development, are there any ongoing pre-competitive consortia more in the discovery phase related to understanding the right targets that you think can be impactful?
Right. So one of the dilemmas we have in pharmaceutical development is high costs, and it takes a long time. And it's obvious here that the processes and approaches, particularly for late-stage studies than the global phase 3 trials, have not been optimized, and all different companies have different systems, and I think it's a pain for the investigators, because they have to train in different systems. In a way, it's the same training, but it's done differently by different companies and so on. So it's a huge inefficiency, and that was the idea then behind the Transcelerate, which came out actually from the Hever group, the heads of R&D, where we identified then a real need for the pharma industry to try to simplify and speed up then clinical development, working together in a very simple way, using common training, common portals, and actually helping the investigators to be more efficient. And we're also working hard, although it's more sensitive, it is to share then comparator material from each other. I personally think it's unfortunate that is sensitive, because we should really share clinical trial material of then drugs that have been approved in a very simple way, and exclude intermediates which just make a lot of money, because we train between ourselves. So that would be also a major cost saving. The need for public-private partnerships are obvious in many diseases, and Mikael and I and some others have recently been very much involved with the National Institute of Health and Francis Collins, and to actually start some new initiatives in the US in some core diseases where I think we all realize that if we join efforts in understanding disease pathophysiology and biomarkers and how we can use them in clinical trials, it would be really beneficial, and we hope that there could be some announcements about that later this year.
Thank you. So I think we have a question over there.
Yeah, thank you for taking my question. Henry Gazet from Medimix. We provide market insight in the field of oncology. In particular, one of the things that we see in biomarker testing is that the US is behind the rest of the world in terms of biomarker testing, which is surprising, but that's a fact. In particular in oncology, where we see rates of around 50% maybe of testing, as opposed to 70 or 90, 70% in Europe, 90% in Japan. So my question to you is, do you think that related to biomarkers, there is a need for better advocacy with the government, certainly, to increase the rate of biomarker testing, or maybe a better relationship between pharmaceutical company and diagnostic testing by either paying for certain biomarkers or really improving the rate of biomarker testing in the country?
You want to start?
I'm not actually aware of the average statistics. I know that in chronic myelogenous leukemia, for example, there's different practice patterns in Europe than the U.S. On where the patients are treated. There are a lot more specialty-oriented centers, whereas the average hematologist in the community will see the patients here in the U.S. So the concentration of the experience leads to different ways of handling and understanding some of the newer technologies. I also think that biomarkers are important to the payer because they tell you which patients not to treat. And I think in some places, the tightening between the regulatory and the payer agencies have led to good medicine and hopefully more cost-effective medicine and greater use of diagnostics. I don't believe, and we looked into this just from one organization's standpoint, that you have to combine with the diagnostic company. I do think, as pharmaceutical companies, we can't just offer a medicine. We have to help facilitate the right type of care and do so with the payers directly in more instances. Jeff, you have?
No.
Thank you.
Any further question here?
Can you hear me? So this is a comment carried over from the prior panel, but also I really wanted to thank the CEO of Vertex for discussing patient advocacy organizations as critical in the ecosystem, but not only for helping to get to patients to enroll them in clinical trials, but there are several of us here and a growing number of us out in the world who are funding research and working directly with pharmaceutical companies, especially in that proof of concept de-risking stage, which is so important to hopefully help tip your balance. So just for the prior panel, just to make that comment that some of these intersections between academia and industry can actually be facilitated and catalyzed by patient advocacy organizations that are highly sophisticated in understanding the need, the unmet medical need, but also the science. So I want to just sort of throw that out for the group.
Thank you.
Can I just make one quick comment? So absolutely agree, and maybe I didn't go into enough detail about this interaction. It was helpful, as you said, from funding research, and is continuing to fund research across many companies in CF, so that was helpful. It was helpful because the patients were really well organized, and so when we got to clinical trials, it made it possible to enroll them really quickly. And you may know the CF Foundation has just done an incredible job of putting these treatment centers together so that these patients receive a very high quality standardized care, which has made an enormous difference for their outcomes, but also makes it a lot easier to do clinical trials when people are getting treated with a standard of care and you can add your medicine on top of it. So that interaction has just been incredible. And it's not only the foundation, it's the patients themselves who are so involved. And many of them come visit Vertex and sort of give our employees pep talks and encourage us. And it's just an incredibly motivating thing when you see these patients come in after they've been treated.
Yeah.
So
I wanted just to punctuate what you have said. We have had fantastic experiences working with cystic fibrosis, lupus, and also melanoma research foundation. And it's really a mix of getting the patient perspectives, understanding the accessibility and inclusion criteria of patients, also helpful funding for research that may be necessary to move a project into a more validated space where we as a company can really apply our resources effectively. And I think really also when it comes to moving the policy dialogue forward, I think Patient Foundation are instrumental. One part that we didn't touch so much on this balance, and I thought would be nice to come to at the end here, is how we feel about how innovation is valued, how it's valued today, and where is the future going towards, and what can we do to influence it. So I'll start by asking Jeff, in the space of orphan disease, patients with little hope gain access to really life-saving medicines. But as we know, obviously the value companies attribute to them are high. And could you reflect a little bit on your experiences with Lilly and what is critical for you to see to continue in this value, how you put value on innovation for other patients with rare diseases to experience the same type of relief?
Yeah, maybe two comments. Kalydeco is a nice example because we treat about 2,000 patients in the U.S. and about 2,000 patients in Europe. So it's a sort of ultra-orphan almost. We really can never talk about value or price without talking about access, because to me this is really about making sure that the patients get the medicines and can get the medicines, and that no patient goes without a medicine because they can't afford it. And so, for instance, in the U.S., we put in place a very aggressive patient assistance program that basically says that if a patient has no insurance, we basically give them the drug. If a patient has insurance but has a large copay that they can't afford, we will supplement that copay down to the level of $10 or $15 a month, sort of what they would pay for their Lipitor, et cetera. And the result of that has been really within 6 to 8 months of approval, we had more than 90% of the G551D patients in the U.S. on drug. And at least I'm not aware of any patients here that have not been able to go on drug because of economic issues. The other comment that I would make, though, is in many ways CF and Vertex being a smaller company, to me is a beautiful example of how the value proposition plays out over time. So we had Kalydeco approved a little over a year ago, and we are using not only all the money we make from Kalydeco, but extra money that we're getting from the foundation and others to develop this next generation of drugs. And we'll have 2 or 3 generations of cystic fibrosis drugs, I hope, that allow us to reach 90% of the patients, and that will essentially be a continuous self-funding effort in which the revenues that we get from these initial small set of patients can be used to develop the set of medicines that will treat the larger sets of patients.
Jan, any reflection from your side on both the opportunity for early invention, the need for regulators to put emphasis on facilitating that process in Alzheimer, and how do you think payer may welcome possibly the first Alzheimer drugs that may have more of an incremental effect for few, but maybe the building block to combination therapy, as you alluded to, that could really add years?
Well, it's very important that we, in the pharma industry, not work in isolation. Clearly, we are here to help patients, and we need really to then align with regulators and payers to be able to do that. And it may be a somewhat different discussion, you know, in the rare diseases versus the common diseases, which is somewhat unfortunate in a way, remembering that the cost of medicines in healthcare usually is said to be about 10% of the total healthcare costs. And a few months ago, I read an article in the Time where they actually describe where do costs come from in the U.S. healthcare system, and I would encourage you to read the article, and I don't want to comment more since I come from the pharma industry, and I think the costs do not come from the pharma industry to any large extent. So which I think means that if we can have innovative medicines with proven differentiation, we should be rewarded as an industry to taking those risks. If you work for 25 years in Alzheimer's disease and spending billions of dollars, you know, you should have at least a hope to getting some reimbursement in the end for the new medicines, particularly if it helps millions of patients then having a better quality of life, and perhaps then aging with a better brain function, which is, I think, something that we all aspire to. I think it's more of a political debate as well, you know, that how do you value quality of life, and how do you value that in a tight economic environment, which I think we realize we have. But I would ask for more transparency about where do the healthcare costs come from in the US, and what is the impact of new medicines that could actually reduce the cost in the end. And at least for me, then, quality of life is something that I personally would be prepared to pay for, and it's really, I think, up to our politicians to step up in this area.
Thank you, John. So let's conclude this session, and I thank you all for taking part, and the panel for your attention. And before we leave the room, I just wanted your eyes to focus on the balance. And as you can see, we as a group here feel that after a decade of a balance in the wrong direction, it has slightly tipped where the positive opportunity driven by science, understanding human disease, pharma, academia, governmental interaction has allowed some force to move us forward. But this balance is delicate, and we need to work together to keep us going in converging all these positive scientific technical factors and try to mitigate the pressures so that we can see this balance when we meet here in 1 or 2 years even more stronger, leaning in the right direction, and obviously hear about many more impactful medicines and start to think in many more areas about treatment advances that are durable, long-term, and maybe even in some cases, complete remissions that last lifelong. Thank you for your attention.